Welcome to the lair of the Batty Boffin.
Hi there, Batty fans. Welcome to another Batty Boffin experiment. This is number 21, Getting Rotten. If you'd like, there's a downloadable project sheet available at this at www.battyboffin.co.uk. Pop along there and print it out totally for free, and it'll tell you all about this experiment. Now, what we're looking at in this experiment here is microbes. Now, what are microbes? Well, it's just a posh word for little things, really. Basically, we're talking bacteria and that kind of stuff. Stuff that's so small you need a microscope to see it. Now, although you can't see these microbes, they are all around us, all over the place. Breathe in. You've just breathed in some microbes. There are millions of them. The vast majority don't do any harm to you at all. Some of them, yes, will make you ill. If you get a cold, that's a microbe that's making you ill there. Some of them, we know, can make your food go rotten. A whole load of them, of course, though, are very, very useful to us. You've got millions and millions of microbes living in your gut, and they digest your food for you. If you didn't have those microbes there, you wouldn't be able to eat your food. In fact, here's something weird. If you counted up all the cells in your body, there are actually far more bacteria cells in your body than there are human cells. Now that's just weird, but you are mostly actually alien bacteria. But they're very, very useful, so don't worry about them, you need them. But we're going to be talking in this experiment about the bacteria that are not so useful, the ones that make your food go rotten. And for this, we are going to need some bread. We're going to need some containers to put it in, either kind of little boxes like that with lids, or you can use bags, doesn't matter, either one of those. And we're going to need some labels as well. Here are my labels. So what I'm going to do with this is just going to start off putting a piece of bread in a box, okay? And I'm going to cover it up like this to stop any airborne microbes getting into it. Now, there's already microbes in there. The bread's got microbes in it. It's just in everything. So that's just normal. This one I'm calling control. Now, every experiment needs to have a control. And the control is the one that you don't do anything to. That's just going to be a piece of bread in a box, and that's all. And then when I do other boxes that are similar, but with something a little bit different, then if there's any difference in the results, I can guess that the something I did different caused the change in the results. But you need to have something to compare it to know what would have happened if you did nothing, so that's the control, the nothing. So that's just my piece of bread in a box, and I'm just going to leave that. I'm going to take photos of it every day or so, and you'll see them later on in this video. So that's my control, number one. For number two, I'm going to do the same thing, but I'm going to soak the bread in vinegar, and we'll see if that makes a difference. The reason I've chosen vinegar is because that's one of the methods of food preservation that we use. Think pickled onions? Yep, that's in vinegar. For the next one is another common method of preservation. Putting things in salt. Things like bacon, they're covered in salt to keep them fresh. So I'm going to try putting one in salt. For the next one, I'm putting it in sugar. Of course, that's things like jam. For the next one, I'm going to toast my bread very, very lightly just to dry it out. And we often have dried food. For example, we get dried noodle, dried rice, dried tea. They're all dried foods. For the next one, not quite so edible, but I'm going to put bleach on my bread, hoping that that will kill all the microbes in the bread. For the next one, I'm going to microwave it. I'm hoping that the microwaves might kill all the bacteria as well. We'll have to see how well that works. We use that on things like when we can goods. They're heated to a very high temperature to kill all the microbes in it, so it's something like that I'm hoping. The next one, I'm going to soak it in water. That's not a normal method for preserving, but I'm just going to see what happens there. Just as a comparison, for number 9, I'm going to lick the bread and see what grows on that. That one could be a bit kind of gross, really. We'll see what happens. Number 10, I'm going to keep it in the dark. Will that make any difference? I don't know. And now some more normal ones. For number 11, I'm going to keep it in the fridge. And for number 12, I'm going to freeze it in the freezer. So there we've got some kind of normal methods of preservation and some slightly weird ones, and some that frankly I don't think are going to work at all. But we'll see what happens. Now, you can choose your own things you want to do. Definitely have a control, and then pick a selection of others. They could be mine, you could do different ones. Make sure you label them up, take photos of them regularly, and watch what happens. So here come my photos of what's been happening. So this is my control, day 2, day 4, day 6 we're starting to get a little mould, day 10, 4, 12 and 14, oh my word. Sample 2, preserved in vinegar, we can see looks absolutely fine. I personally wouldn't want to eat it, but it's beautifully preserved. The salt sample, similarly, the bread seems to have dried out. You can see it curling at the edges, and again, beautifully preserved, but I wouldn't really want to eat it. Mind you, works fine for gammon. Sample 4 is preserved in sugar. This one actually probably would taste quite nice. Again, you can see it's working by drying. The bread is curling here, and I guess it would just taste like bread and honey. Sample 5 has been dried by very lightly being toasted, and again that has worked beautifully. Obviously the bacteria can't grow in these dry conditions. Great work. Now sample 6, I poured bleach on it, and to be honest I thought it would do better than this. Clearly not all of it got soaked in bleach and left a good clear run for the nasty bacteria to grow. Sample 7 I microwaved, hoping that that would kind of kill the bacteria by cooking them, and it seems to have done a pretty good job. That was a beautifully preserved sample, and I'd eat that piece of bread no problem. On the other hand, the piece of bread that was put in water, you can see within a day it had started going horrible, and it stank to high heaven. There's nasty stuff growing on this. Sample 9 I licked. I didn't really think that would preserve anything. I just wanted to see what kind of things would grow. This one also smelt pretty bad, but not as bad as the one in water. I kept sample 10 in the dark to see if that would do anything, and really it didn't make any difference at all. This is pretty much the same as the control, which shows the bacteria do not require light to grow. Sample 11 was kept in the fridge, pretty normal means of preservation, and yeah, that piece of bread is absolutely fine. Give it a month or so, it'll probably have grown some bacteria, but within the 2 weeks of this experiment, it was fine. Sample 12 kept in the freezer, as expected, is absolutely fine, not a problem at all. I've had a lot of fun doing all this here, apart from the smelly stuff. I hope you have too. I'll see you next time, Batty fans.
Mwah! Mwah! Mwah!